Hello students, welcome in Babita Science tutorial. Today let's start our new chapter, that is a chapter 5, and its name is the fundamental unit of a life. Okay students, and the fundamental unit of a life is belongs from your biology. Okay, and this is our new chapter of a class 9 science NCERT. Actually, basically in this chapter we are study about the cell. Okay, the fundamental unit of a life. Then our fundamental unit of life is a cell. So in this chapter we will study about only the cell. Let's continue our chapter here. Now see here, while examining a thin slice of a cork, okay, Robert Hooke. Robert Hooke was a scientist who was examining a thin slice of a cork. Student, what is a cork? Cork is a substance which comes from the bark of a tree. Okay, and what is a bark, student? Suppose that this is your this one is a tree. Okay, and the outer covering of this tree, okay, here, that is called your bark. Then cork is comes from the bark. Okay, then Robert Hooke was examining the cork, and he found that the cork resembled the structure of a honeycomb consisting of many little compartments. Okay, little compartments means honeycomb means madhumakhi ka chatta. Okay, he found that cork is just like a honeycomb and it has many little compartments. Little compartments means like this. Okay, hexagonal shape like this. Okay, this is a many little compartment. He found that the structure of a cork is just like a honeycomb, and he was discovered this one in 1665 when the Hooke made this chance observation through a self-designed microscope. Means Robert Hooke was discovered this cell in 1665 with a self-designed microscope. Okay, then Robert Hooke called this boxes cell, and cell in Latin word for the little room. In the Latin word, the meaning of the cells is a little room. So in this paragraph only they want to say that Robert Hooke was discovered the cell with the help of cork and he was using a self-designed microscope, and he was discovered the cell in 1665, and the meaning of the cell in the Latin words is a little room. Now in this paragraph they want to say that a small and insignificant incident but is a very important in the history of a cell. Means they want to say that discovery of the cell is a very important in the history of a science. Okay, and this was the very first time that someone had observed that living things appears to consist of a separate unit, and in this time they want to see that someone had discovered, means Robert Hooke had discovered that the living things consist of separate units. Okay, and these separate units in the biology is your cell. Then we can say that Robert Hooke's discovery was important because it indicated for the first time that living organisms consist of a number of small structures or units. Okay student, and here units means a cell. So in this paragraph they are discovering cells in the year 1665. Meaning of the cell is given here, little room, and they want to say that it was very important in the history of a science discovery of the cells. Let's move in another paragraph. Now, here is a 5.1. What are living organisms made up of? The students, with the help of this activity, we can understand what are the living organisms made up of. We know, I know, you know also what are the living organisms made up of. Very simple. Living organisms are made up of by cells. Okay, but with the help of this activity we can understand it. Now in this activities, take a small piece from an onion. Okay, onion bulb with the help of the pair of forceps, and peel off the skin called the epidermis from the concave side inner layer of the onion. This layers can be put immediately in the watch glass here. Okay, watch glass containing water. This will prevent the peel from the getting folders or getting dry. What do we do with this peel? Students, first of all, I want to explain this point. Okay, we will not move very fastly, we will go slowly, then you will students understand better here. They want to say that take the peel of a onion. Okay, this is the onion. Okay, when we remove the outer layer of the onion, then you will take a thin slice white color layer is present on the inner sides of the onion. Okay, and take this thin layer of this onion. Okay, and see here inner of the onion, and this layers can be put immediately in a watch glass. Okay, now this is your watch glass, or we can use the word petri dish also. Petri dish also, and with the help of then students, you will take this thin slice in this petri dish. Okay, thin slice of a onion in this petri dish, and in this petri dish is a water is present. Then question is that why we keep this thin slice of a onion peel in the water? Main is that we can say that we can keep the thin slice of a onion peel in the water to prevent the peel from getting folded or getting dry. If anyone is asking the question to you after removing the peels of the onion, and we will keep inside the batter dish in which water is present, why we will keep the peel of onion in the batter dish? Then you will give the answer to prevent from drying and getting folded. Okay, suppose that this is a peel, then this peel maybe sometimes what happen folded inside it. No, then if we will keep this peel inside the water, then it will not be dry. Okay, and it will not be folded. Okay, that's why we will keep the peel of a onion inside the batter dish. Okay, now see here in the second point, let us take a glass slide. Okay, put a drop of water on it and transfer a small piece of a peel from the watch glass. Okay, students, here they want to say take the glass slide. This one is a glass slide. Okay, it is easily available in the science laboratory, and here is a batter dish. Okay, and in here is a water, and your peel is here. Then put a few drops of a water here. Okay, with a few drops of a water here, and then you will remove this peel with the help of the paint brush. Okay, with the help of the paint, a thin camel hair paint brush. With the help of that brush, you can shift this peel in this slide, or you will keep this peel here. Okay, And then after we will put a few drops of a saffron in solution. Okay, we put the saffron in solution. The saffron in solution is a pinkish in color. Okay, you will keep the here saffron in solution and put a cover slip. Students, cover slip is comes in the shape of the square also and round also. Then cover slip, it is also transparent and made up of the glass. You will keep this cover slip on this slides with the very slowly. Okay, we will keep this cover slip on this slide very slowly with the help of the pencil or with the help of the needle, and take care that the bubbles will not come inside this slides. Then what happened here? Avoid air bubbles while putting the cover slip with the help of the mounting needles. Okay, I told to you mounting needles means it's the one kind of the needle which we use in the lab. This is a pointed and the back portion is a broad. Okay, with the help of this needle you can put the cover slip on the slide. Understand? That's the meaning of the mounting needle. And why we use this needle to put the cover slip? To prevent air bubbles. Okay, students, suppose that this is your slides and peel is here, saffron solution is here, then cover slip is here. If lots of air bubbles will comes under this slide, the when will observe these slides under the microscope, then what happen? You will see only the bubbles, big bubbles under the microscope. You will not see the cells. That's why we can take the precautions while putting the cover slip on the slides. Okay, students, that's why we will take the precaution here. Now here, ask your teachers for help. We have prepared a temporary mount on NP. We can observe these slides under the low power followed by the high power with the high powers of a compound microscope. Students, see here temporary mount. Okay, on NP. Here is the two types of the slide. One is the permanent slides and another is a temporary mount. The temporary mount means those slides which is made by the students recently or on the spots or in the laboratory. That is called the temporary mount. Or if any slide is made permanently, that is called the permanent slide. After that you will see this slide under the microscope. Student, this is your compound microscope. You will see here that this structure is a very important compound microscope. The upper one is called the eyepiece, then body tube, coarse adjustment, fine adjustment, arm, objective lens, stage, then civil mirror base is given here, microscope, slides, condenser is given. Okay, student, now see here, this one is also called the light microscope. Here eyepiece is given here. Okay, this one is the eyepiece. Then from the eyepiece on the top one can see a magnified image of the object or specimen. Okay, with the help of this eyepiece we can see the object. Okay, this one is your body tube. Okay, we can keep the slide here, and here you will see there is a two adjustment here. First one is a coarse adjustment and another is here fine adjustment. The upper and large knob is your coarse adjustment, and it is used for the rapid and accurate focusing on the object. Okay, coarse adjustment we can use for the accurate focusing of the object, and the lower one is called the fine adjustment for getting perfect image of the object. Fine adjustment we can use for the perfect image of the object. Okay, now see here mirror is present here, and a condenser is also present here. Then light is passed through the object or specimen with the help of the mirror. This one with the help of the light is passed to the object with the help of the mirror, and here is a condenser is given here. So this is your compound microscope. Then students, you will when you will keep these slides under this or when you will observe these slides under this microscope, the which type of structure you will find here. Then you will see the structure like this. Okay, then see here, what do you observe as we look through the lens? Can we draw the structures that are able to see through the microscope of on an observation sheet? Does it look like? Now see, yes, this is a cell of an onion peel. See here, this is called the small compartments, and the cell structures are also here different. So this is this one is a very important in the practical also. Okay, and for the diagram point of view also, draw the cells of an onion peel, student. Then this is all about of this page. Then what is important here? The importance is only this, that who discovered the cell in which year, okay, and from the which substance Robert Hood discovered this cells. What is the Latin words meaning of the cells? Then here experiment of a onion peel. Okay, experiment of a onion peel, structure of a onion peel, and the diagram of this compound microscope. This one is a very important here. So student, this much only about this page. You can read this page thoroughly and understand it, and if you problem, then comment me in my comments. Till then, bye, have a nice day.